So we're going to come back to this today. We're going to see this again, but you're not going to see this unfolding the way that you're going to see it unfolding right here. And what I hope is that this kind of helps you understand a couple of things. One is that I think will help you understand the complexity of what's going on, the degree to which vocabulary is critical. The other thing is I hope that it kind of helps you picture what the various bits and pieces that we're going to be talking about look like. So let's start with a piece of DNA, okay? And as you know, DNA is typically double-stranded. So if we have one sugar phosphate backbone like this with a bunch of A's and C's and G's and T's on it, yeah. Yeah, right over there, yeah. It will also have another complementary sugar phosphate backbone, and all the bases on there, they won't be the same, right? If one has an A, the other will have a T. If one has a G, the other will have a C, but they'll be complementary. So while they're not the same, they're sort of mirror image. If you have one of these, you can figure out what the other one would be. That's what you did with your DNA models. So they're both there, and they both have different sequence of nucleotides on them, but the sequence is predictable. If you know one, you'll know what the other one is going to be, because it's always going to be the complementary base pair. So if this one has A, C, G, G, then the green one will have T, G, C, C, and it'll always work like that. When DNA replicates, which is what we talked about last week and a little bit this week, what happens is that these strands separate, and they separate in various places depending on what kind of an organism it is, but we typically sort of show it separating, on one end, like the bottom or the top. And so both of those strands are still there, but now only part of them are intertwined, and the other part of them are separated and single-stranded. And when DNA replicates, what happens is we build a new complementary strand here and a new complementary strand here. And ultimately, the whole thing pulls apart, and we get another complementary strand on each of our original parts of the first piece of double-stranded DNA. But it's kind of weird, because how many pieces of DNA are we looking at here? We could say that this is one chunk of DNA, but it's double-stranded, so it's kind of one chunk that's made of two parts. Are those two parts the same? Well, no, but they're complementary. And over here, what do we have? Well, it's kind of one thing, because it's all attached. Like if I were to pick this up with tweezers, like it would all stick together. But also, it's kind of two things, like here's one thing here and here's another thing here, but it's also kind of like, now there's like another, like a third and a fourth. Like how do we even count this? Like what is this thing that we're looking at, and how many parts does it have? It gets a little confusing. If we were to think about the bases on here, if this is A and this is G and this is C, that would make this over here a T, this would be a C, this would be a G, and these bases, I have them pointing the wrong direction just so I have room to write, but obviously they would be connected between the strands of DNA. And so that means like all we need is one of these to get the information. You give me one strand of DNA and I could build the other strand. And then if you take those two strands and separate them, we're going to find the same thing. If this strand over here had a C and a C and an A, then the new strand that's built will have a G and a G and a T, but the old green part of the strand also had a G and a G and a T, And the new red part will also have a C and a C and an A. So what do we have here? Like, how many pieces are there? And it gets even more confusing because when DNA replication is done, we don't actually end up with two physically separate double-stranded chunks of DNA. Instead, they stick together. So what we would end up with is something that looks like this. Here's our original red strand with an A and a G, that was the world's worst A, with an A and a G and a C and a C and a C and an A. That's our original red strand. Here's our original green strand. It would have a T, a C, and a G, and it would have a G, and a G, and a T. But we also have a new green strand along here, and that one's going to have a T, a C, a G, a G, a G, and a T. And we have a new red strand along here, and that one's going to have an A, and a G, and a C, and a C, and a C, and an A. So all the red strands are the same, and all the green strands are the same. This original double-stranded chunk of DNA is the same as this new chunk of double-stranded DNA, one piece of which came from the original, and one piece of which was added on. It's also the same as this chunk of double-stranded DNA, one piece of which was the original, and one piece of which was newly added on, and they're connected. They're connected generally somewhere near the center. And what ends up happening is we start with one piece of double-stranded DNA that consists of two complementary single strands of DNA. We end up with four complementary strands of DNA. These two are complementary, these two are complementary, these two are the same as these two, but they're hooked together in the center. This is what happens after DNA replicates. And the whole point of DNA replication, we talked about the other day, was to enable cell division. So ultimately what happens is you start with one of these in a cell, one single double strand of DNA in a cell. It gets replicated, and then one chunk of DNA goes to one cell, the other chunk of DNA goes to another cell. And we end up with two cells where we had one. Both new cells have DNA that is identical to the original DNA that was in the first cell. This is what we're going to be looking at in more detail today. What in specific we're going to be looking at is what happens to these things. These pieces of DNA where there is part of the original strand here, the other part of the original strand here, a new strand here, and a new strand here. We essentially have two chunks of double-stranded DNA that could kind of live on their own. But at first, they're stuck together. And mitosis is how this DNA gets neatly separated so that inside new cells, they have all the DNA that they need. They have all the DNA that the original cell had. Okay? So we'll come back to this diagram down the road because what you're going to see is that it looks and feels a little odd as we start like thinking about, well, sometimes DNA looks like this and sometimes it looks like this, and like how does that happen? This is how it happens. We start here and then it gradually looks like this. I mean, that process happens pretty quickly, but it's not instantaneous. And then something happens here to make us go back to this, and that something is mitosis and cell division. Okay, so our goals today are for you to be able to think about and talk about the cell cycle, and to think about the process of mitosis, which is only part of the cell cycle, and then to be able to think about why we even need that. Like, what is the point of having cells divide? Why do we need cell division? Why do we need mitosis as part of cell division? Okay. So the big picture here is that all cells at some point undergo what we call the cell cycle, and mitosis is just part of the cell cycle. So the cell cycle is kind of like the process that a cell goes through as like, it's just sort of what cells are all about, right? So the things that go on in a cell, that the function that the cell performs is not really part of the cell cycle. The cell cycle has to do with how cells divide. And mitosis, which is what we're going to focus on today, is just part of the cell cycle. So in order to divide, cells need to do two main things. One, they need to grow, and they need to duplicate stuff. So if I have a cookbook with some recipes in it, like breakfast recipes, and Jacob comes to me and he says, Brigar, I've heard that you are the breakfast king. You make the killerest breakfasts in Corvallis. I want your recipes. I'm not going to give him my book. Because if I give him my book, then I don't have my book. And how am I supposed to make my awesome breakfasts if I don't have my book? So before I can confer this information on somebody else, I have to make a copy of it. And that is kind of what happens in cells in general. They have to make copies of everything. New cells need to have mitochondria and ribosomes and endoplasmic reticulum and lysosomes and Golgi apparatuses. And so when a cell is going to divide, it has to get bigger, and it has to make copies of all those things. And then the cell actually has to physically divide. And when it physically divides, there's two parts to that. One part of that is splitting up the DNA equally, and one part of that is splitting up everything else. And so we'll talk about mitosis, that's the DNA splitting up part, and then cytokinesis is the rest of it, okay? And that together is known as the M phase. And on this diagram right here, which shows the cell cycle, this part right here is the M phase. It consists of mitosis, which we'll talk about more, that's the division of DNA, and cytokinesis, which we'll talk about more, that's the rest of the cell getting split up and physically divided. The rest of this time, as we'll see in a second, is known as interphase. That's where everything grows and gets duplicated. Oops. So these symbols here, G1, S, G2, they're really actually, you don't need to know this, but just for those of you who like to have a word, this just means growth. G is for growth. S is for synthesis, and in particular, DNA synthesis. So during G1, the cellular contents are duplicated, not the DNA, but everything else, and then the cell gets bigger and bigger and bigger and bigger. During the S phase, this is when replication occurs. The DNA is synthesized, it's copied. The thing that I just showed you, this stuff, that happens during the S phase. This is what's going on during the S phase. You go from single double-stranded pieces of DNA to double-stranded pieces of DNA. And then this G2 is just some more growth, and there's some error checking that goes on there, but you don't need to know the details of these things. You don't need to know exactly what happens. Just the idea is that the cell has to grow and duplicate stuff, and then it all gets split up. First the DNA gets split up, and then everything else gets split up. This interphase piece, which we'll just talk about super briefly, it happens before mitosis. And basically this is where the cell divides by getting bigger and duplicating things. Okay, so these G1, S, G2, which is growing, replicating DNA, growing some more, is altogether known as interphase. Inter means between, and what we're talking about is between cell divisions. This is all necessary for cells to divide. We don't think of it as part of the actual process of dividing cells. Mitosis is the division of DNA. So during mitosis, what happens is identical copies of DNA are prepared for each cell. And we'll talk about some of this stuff in a little bit more detail in a second. But basically, inside cells, there's not just one piece of DNA. There's a bunch of pieces of DNA. And those pieces of DNA are called chromosomes. You've heard of chromosomes before in other science classes. I've mentioned the word in here almost certainly. Those chromosomes are physical things, chunks of DNA. Each one of these blobs here is a chromosome. That's one, that's one, that's one, that's one. And when chromosomes when chromosomes replicate, they make this X shape. So this is one, this is one, this is one, and this is one. If we go back to my diagram that I drew here, this is one chromosome. It's a double-stranded chunk of DNA. As long as the DNA is physically attached, it continues to be referred to as one chromosome, which means as this DNA is replicating, this is also one chromosome. And this is also one chromosome. And that seems weird because there's more here and even more here. Why are they all one chromosome? And the answer is because when scientists were first looking at these things, they had no idea what they were. They didn't know it was DNA, and even if they had known it was DNA, they didn't know what DNA was for. They just saw stuff. This is one thing. This is one thing with a sort of a Y shape. This is one thing with sort of an X shape. They had no idea what they were looking at, so they just called it all one thing. It turns out now we know that this is one thing that's made of two things, two strands hooked together. This is one thing that's made of like 2.5 or 3 things. Two strands that are hooked together in some places, but also coming apart, and new strands are being built. This is one thing that's made up of 4 things. 2 single strands of DNA hooked together here, 2 single strands of DNA hooked together here. These 2 strands are identical to these 2 strands. We didn't know that at first. We just saw blobs. So we called those blobs chromosomes, and we'll come back to that in a bit. Yeah?
Are those like supposed to be like X and Y chromosomes?
No, but that's a great question. They're not called X and Y chromosomes. There are in many organisms X and Y chromosomes. They're not named for their shape. It is a coincidence that the X chromosome is X-shaped. In fact, all replicated chromosomes generally either look like an X or sometimes a V. There's not really any chromosomes that look like a Y. The X chromosome was named the X chromosome because it was unknown what it was all about. And X is kind of a symbol for the unknown. So that's where that came from. Confusing, huh? Isn't that terrific? Don't you kind of wish scientists would just start over and do all this right? But they won't. Okay, when mitosis occurs, and we'll come back to this, so you can write this down, but when we come back to it, you'll see some diagrams and stuff. When DNA is replicated, those chromosomes, when they're in this X form, are composed of what we call sister chromatids. So just for now, just write that down. We'll come back to it. We've got more diagrams to come that will hopefully help you understand this. And what mitosis does is it splits up the sister chromatids so that each new cell gets one full copy of the DNA. Here's how I like to think about this. I like to think about it like cookbooks. So when I think about chromosomes, I imagine that one of those chromosomes has recipes for breakfast. So there's like a breakfast book. Whoa, that wasn't at all what I wanted. I hate this new system. It's really the worst in some ways. Okay, let's see if I can do this right this time. So there's, no. Ah! I'm gonna throw a temper tantrum. You guys okay with that?
Yeah.
Why? Resume slideshow. Just, I don't know why this isn't working. Okay, let's try it. All right, okay, good. Breakfast book. So we've got a breakfast cookbook, and that would be one chromosome. And then maybe another chromosome is desserts. So you have a dessert cookbook. And then maybe you have a dinner cookbook. And these actual physical books have instructions in them. So let's say, now these aren't cookbooks, but this will kind of do the same sort of job. We have some books back here. Let's say that these this is my breakfast cookbook, right? So if I want to have everything, I need one of these. I need my breakfast cookbook, and then I need my dinner cookbook, and then I also need my dessert cookbook. So I need 3 cookbooks. So when DNA gets replicated, we make another copy of each of these. So I take my dessert cookbook and I make a copy of it, and it's sort of attached, right? So they're not quite separate yet. They're sort of attached like this. So now I have 2 dessert cookbooks that are physically attached to each other. And I do the same thing with my breakfast cookbooks. Make a full copy, but it sort of stays attached. And make a full copy, and it sort of stays attached. So these are stuck together, but it's really 2 copies. And then I do the same thing with my breakfast cookbook. So I've got my breakfast cookbook, my dinner cookbook, got my dessert cookbook, and I've made copies of each one. Now, if Sebastian and I want to each have a restaurant, I can't just be like, well, there's 6 books here, so here, Sebastian, you take these 3. Because now Sebastian has 2 breakfast cookbooks and 1 dessert cookbook, and I have 2 dinner cookbooks and 1 dessert cookbook. Neither one of us has everything we need. When we make copies of this, we have to make sure that we each take 1 of each. We each need 1 of these, we each need 1 of these, and we each need 1 of these. So we have to divide it up carefully. That's what mitosis does. It divides the chromosomes up so that each new cell gets 1 copy of each chromosome. And mitosis, so we're not gonna have to worry about this too much now, but when it comes to organisms reproducing, sometimes we need a slightly different type of division. We need to do things a little bit differently. That little bit different we'll look at after spring break. It's called meiosis. What we're looking at is how individual cells make perfect copies of themselves. That involves this process of dividing DNA up called mitosis. When reproduction is necessary for sexual reproduction, we need something slightly different. You don't need to worry about that for now. Just kind of pack it away in the back of your mind for future reference. Okay, so I have told you a little bit about this already, and this is where I hope those diagrams that I drew at the start are helpful, because when you look at these images, what we're seeing here are replicated chromosomes, but what we're missing here is that this is a double-stranded piece of DNA, and so is this. And this is a double-stranded piece of DNA hooked to another double-stranded piece of DNA, and this is a double-stranded piece of DNA hooked to another double-stranded piece of DNA. We don't see that in these diagrams, but that is what we're actually looking at. So when you start working with the assignment today, keep that in mind. So most cells have more than 1 chunk of DNA. And each chunk of DNA is actually two pieces, two strands that are complementary to each other, hooked together. Each of those chunks is what's called a chromosome. And I told you that when people first saw these, they didn't know what they were. They just saw these blobs. And here's the thing about chromosomes is that you can't, if you just look at a cell, you won't see them. If you put some dye in a cell, certain kinds of dyes, sometimes you can see them and sometimes you can't. You can see them during certain parts of the cell cycle. During the M phase of the cell cycle, the mitosis and cytokinesis part of the cell cycle, you can see chromosomes because they get physically smaller. And if you've dyed them, that dye becomes concentrated enough that you can see it. Much of the time, when you're looking at chromosomal material, even in a dyed cell, it just looks like this. Just like maybe a faint hint of color. But at the right time, those chromosomes are smaller, that dye is more concentrated, and we see these blobs. You can probably see from where you're sitting, these are X-shaped blobs. There's one X, there's one X, there's one X, there's one X. This one looks more like a V, and so does this one. There's an X. And that's what people saw in their microscopes. They had no idea what they were looking at, but they knew they were colored. That's where the chromo part comes from. And they knew they were blobs. That's where the some part comes from. In Greek, chroma means colored, some means body. These are named colored bodies. We didn't know what they were, but we knew that they were colored blobs. So some genius was like, I know, let's call them colored blobs. Only colored blobs sounds like something a kid would come up with. Let's use Greek, because that makes it sound fancy and scientific, and that is where the name chromosome came from. Typically, when you look under a microscope at a condensed, unreplicated chromosome, it kind of looks like a little sausage. Diagrammatically, we typically will draw it like this. This circular blob here is not visible. We don't see that easily through a light microscope. We actually, you can see it a little bit, but it doesn't look like this. This is sort of schematic. What's really going on here, this green thing right there, is the equivalent of this chromosome right here. There's two strands of DNA that are complementary, but there's only one set of those two strands. That would be an unreplicated chromosome. And then when the chromosomes replicate, most of the time they look like an X. Sometimes they kind of look like a V. There's no meaning to the X or the V, it's just where they're attached. If they're attached in the middle or closer to the middle, they look more like an X, like this one and this one. If they're attached closer to the ends, like this one and this one, they look more like a V, and that's it. That's the only difference between the X-shaped and the more V-shaped chromosomes. And again, in my diagram, that's this. You can see the X here. This is one double strand of DNA physically attached to another double strand of DNA. At this point right here, it makes an X because it's physically attached in the middle. At this point, this is still referred to as one chromosome. And that's just by convention, because again, when people looked through the microscope, what they saw was this. They didn't know that it was two identical things attached in the middle. They just knew that it was a blob that stuck together, so they called it a chromosome. And then when DNA is replicated, each individual piece is called a sister chromatid. Now, if we were going to do all this over again, probably we would not use this same vocabulary. Probably we would call each replicated piece a chromosome. Like, it makes more sense to say, this is one chromosome here, this is another chromosome here. This is two chromosomes attached together, and this is two chromosomes attached together. Probably that's what we would do if we were to start over, but it's too late. So instead, we call this one chromosome that consists of two identical sister chromatids. This is one chromosome that consists of two identical sister chromatids. When we get to meiosis, we'll talk about homologous chromosomes because it gets even more confusing, but for now, you don't need to worry about that. So here's one chromosome, here's another chromosome. Here is one chromosome consisting of two identical sister chromatids. Here's another chromosome consisting of identical sister chromatids. When you look at my diagram, these are the sister chromatids. This is one sister chromatid here, and here's the other sister chromatid here. Each sister chromatid consists of 2 strands of DNA that are complementary to each other, hooked together in a double helix. This sister chromatid is identical genetically to this sister chromatid. They have the same information. It's our 2 cookbooks that are physically hooked together. So whichever one this was, I don't remember whether this was, and I know I was getting all confused while I was talking, but whatever one these are, they're hooked together. Same book, hooked together. At this point in DNA language, this would technically be called one book. We all know it's 2, but we call it one. And instead of saying, well, this is one book hooked to another book, what we say is, these are sister booklets, hooked together to make one book. But of course, as soon as we pull them apart, yoink, now it's 2 books. And that is exactly how mitosis works. When mitosis separates sister chromatids, they become individual chromosomes. All right, so here's how mitosis unfolds. There are, the names of these things aren't critical, but you'll see them referred to, and so we'll talk about them. So the first thing to remember is mitosis starts after replication has already occurred. So our chromosomes at the beginning of mitosis are already X shapes of sister chromatids. You can see that here. This is one chromosome, this is another one. This chromosome here consists of this sister chromatid physically attached to this identical sister chromatid. This chromosome consists of this sister chromatid physically attached to this sister chromatid. Okay? During prophase, we start to be able to see the chromosomes. They become smaller and become visible in a light microscope under stain. In metaphase, our chromosomes line up along the middle of the cell, and they line up in a way such that each sister chromatid is on one side or the other of the cell. So this is the same chromosome that we saw before, lined up so that one sister chromatid is on the left side, one sister chromatid is on the right side. This chromosome lines up below it with one sister chromatid on the left side, one sister chromatid on the right side. Then those sister chromatids get pulled apart. So here's one sister chromatid, here's another one. For this blue chromosome, here's one sister chromatid, here's another one for this red chromosome. And the fact that they look like little V's here, and there's like the V is pointing toward the side, is not meaningful. It's not like there's no, it's not like this is fundamentally different. It's just they are getting physically pulled around, and they get pulled around by that center point. So if you imagine some part of the cell, some structure, grabbing onto this chromosome right here, pulling in 2 separate directions, and then continuing to pull, you can imagine that the chromosome would kind of bend and pull like this, and that's exactly what happens. Yeah?
What's with the line inside the metaphase and anaphase? So
These are physical structures called spindle fibers. You don't need to know what that is. Those spindle fibers are what actually physically pull them apart, right? Because it literally has to happen. You have to physically pull these things apart in order for them to move. Otherwise, they wouldn't go anywhere. And then once the sister chromatids have separated from each other, a nuclear envelope forms around each one, and you start the process of dividing this formerly one cell into 2 cells, each of which has an unreplicated copy of each chromosome in it. So what mitosis does is it divides up DNA in a cell so that that cell can split. And it does it in such a way that each new cell will have one copy of each chromosome. Those sister chromatids get split up. Okay? And it's a physical process And that's why these lines are showing the agents of force, like those are actually physical connections from the edges of the cell pulling on the DNA so that it actually breaks apart right in the middle. One sister chromatid goes one way, the identical sister chromatid goes the other way. And so it separates those sister chromatids. It separates the sister chromatids that were created when the DNA replicated. And remember that this is part of that whole thing that we call the M phase, but it's not the whole thing. There's one last piece, which is called cytokinesis. And in cytokinesis, which happens after mitosis, so cytokinesis is what happens after mitosis has taken place. After the DNA has divided, cytokinesis divides up the rest of the cellular contents. So cytokinesis makes sure that each new cell has all the mitochondria that it needs, and all the ribosomes, and the Golgi apparatus, and the lysosomes, and all those other little pieces that have to be in a cell. That all has to get divided up too. And then a new cell membrane and or cell wall are built, and that formerly what was one cell is now physically two separate cells. So cytokinesis is the physical property of one cell dividing into two. Mitosis is the splitting of DNA, physically separating pieces of DNA so that there's a set of chromosomes on one side, a set of identical chromosomes on the other side. And then cytokinesis is how that all happens with the whole cell. If we were to look at this, and cytokinesis is still part of that M phase. So M phase is mitosis plus cytokinesis. If we were to look at this under a microscope, here's what we'd see. During prophase, we just see a bunch of blobs. And those blobs are chromosomes, but we can't distinguish between them. They are now small enough that the stain would be visible, but we can't distinguish between individual chromosomes. During metaphase, we actually see them physically lining up along the center of the cell. The diagram that I showed you before had a left and a right. In this case, the center of the cell is here. Those spindle fibers are coming up like this. This is metaphase. And this is where the sister chromatids are on opposite sides of the cell, but are still attached. During anaphase, the sister chromatids have separated. So now there's a single-stranded DNA down here, or sorry, single chunks of double-stranded DNA down here, single chunks of double-stranded DNA not replicated up there, physically separated from each other. These were chromosomes consisting of two sister chromatids. Now, these are chromosomes consisting of one piece of chromosomal material, and these are identical. And then during telophase and cytokinesis, we start to see, I did that last time too, telophase plus cytokinesis. This is where we see the new cell membrane and or cell wall form, and we have now gone from one cell to two cells. Okay, so for those of you who need more, I'm not going to show this, but there's a included in today's notes is a link to an Amoeba Sisters video. We're not going to play that. What I want to do now is take 5, and then I'm going to have you guys play around with this stuff. So will you come back at 9:03, and I'll tell you what the next steps are?